This is Cabbage Tree Bay Aquatic Reserve, a marine protected area in Manly, Sydney. Stretching from the borders of Manly Beach to just beyond the headlands of Shelley Beach, this small area is a home and rest station to many marine species. Established as a no-take aquatic reserve in 2002, only passive activities are permitted, but this little aquatic reserve will play a big part of the project, aiming to investigate the role of marine protected areas in captive breeding programs in restocking wild populations, in particular the Wobbegong shark. The Wobbegong project was started in 2008 by shark ecologist Dr. Charlie Hooverneers and is now headed by Kate Lee from Macquarie University. Since 2008, 17 captive-bred Wobbegong sharks have been released in Cabbage Tree Bay at Shelley Beach, and their movements will be compared to the 13 wild Wobbegongs that were caught, tagged and released in 2008 and 2009. Cabbage Tree Bay is known for its marine diversity, habitats and conservation value. Dave Thomas has been diving in the area since 1991 and formed his own marine conservation group, Eco Divers. They do cleanups, habitat preservation, as well as the spontaneous animal rescue. He got involved with the Wobbegongs before the project even started, where he took flank shots to show spot patterns and get an idea of the population. He now helps Kate with the surveying of the sharks.
It's what we call like a rest station. We've got a lot of migratory animals come and stay here. We get turtles and dolphins and whales, seals, penguins, a lot of animals that won't normally live here, but they come through and they'll stop here because it's such a protected area. And as you see, it's sort of, that's time, most of the time it's pretty calm, so they'll come in and stay here. Then you have resident animals as well. So from a resident animal point of view, it's very good at protecting them because hopefully there's still fishing here, but it's not as often as it was. So they get to grow up from juvenile through to adult, through to breeding and continue to cycle. Migratory species get to rest here, but then they go on and they're subject to all the fishing pressures and shark nets and everything else that's elsewhere, but at least they get somewhere they can stop. So ideally a linked series of these parks at these places, like similar to this, would form a much better conservation value than odd ones sort of along the way.
Wobbegongs are one such migratory species. They are bottom-dwelling sharks and rarely move when divers are about, but will bite if provoked. Population numbers have depleted from overfishing and shark nets. They are listed by the IUCN Red List as vulnerable. Limitations on fishing have been put in place to manage numbers, and although recreational fishing is banned, the sharks are still fished commercially. The shark pups are bred in Sydney Aquarium and later transferred to Manly Ocean World before their release. They are being released at different sizes to find the optimal age for future releases. They are being tracked using acoustic telemetry technology from the Australian Acoustic Tracking and Monitoring System, a facility within the Integrated Marine Observing System. We speak to Andrew Boom about how it all works at the Sydney Institute of Marine Science.
The easiest way to explain it is if you look and think about your computer modem that you used to have when you had dial-up modem, and it would basically transmit some sound, and at the other end it would get reinterpreted into a signal. And the acoustic tags, that's a very large acoustic tag for a very large animal, and what it actually does is it produces a series of beeps. And so it has, these tags have 8 beeps, and it listens for the beeps and the gaps in between the beeps. And so we put these inside the animals, externally or internally inside the animals. And when they go past one of the receivers, which is this unit here, the receiver picks up the signal, and if you look really closely, all going well, you can actually see the signals being picked up on this unit. So when it's flashing like that, it's just normal, and nothing ever works to cue. That's actually a tag getting detected there.
The positions of the sharks are calculated through triangulation of the 9 receivers that are in the bay and the Vemcombe Positioning System, or VPS. This tracks down the shark's movement in the area to the metre, making it a very fine-scale project.
Looking at these little red dots as the receivers. And what it actually does is, I was saying before, it actually enables you to actually triangulate between where the detections are. So these are the actual detections that they're getting from the receivers. And if you look at this in terms of habitat, this is reef, sandy reef, hard reef, kelpie weed, straight sand. It enables you to determine habitat selection of the animals based on its individual movements. So you can start to get an idea within this, because this is actually a little sanctuary, and you get the idea of how effective this sanctuary is in actually protecting this species and what type of habitat they need.
In charge of the project is Kate Lee, who has just finished attending an international conference where she talks to us from Cairns.
We're hoping by the end of this year we will sit down and we'll completely evaluate all these releases, and we're looking really at the long-term retention. So we're looking to see how many individuals will stay within the area on a long-term basis. Wild Wobbegongs do come and go. Some of them will stay there for 9 months and then leave and return. They can return after a 6-month absence. So we're just trying to see how much these released animals' behaviour reflects the wild ones.
Preliminary results have shown that the captive-bred sharks are assimilating as they have left the reserve at some point, reflecting the behaviour of their wild counterparts.
It means that they're doing the same as the wild Wobbegongs do, so I take that as a good sign. Like I said, we really are evaluating this on a long-term basis. So for the ones that were released in 2008, say after 2 years, how many have done exactly what the wild Wobbegongs will do? That's what we're kind of really asking with it.
Final findings of the project are still a while way off. Further data collection and analysis is needed until final conclusions can be made about the effectiveness of captive breeding in marine protected areas as a restocking method.